Okay, we'll go ahead and get started. One announcement, you have your exam 5A opens today at 2:15, closes tomorrow at 2:15. You'll have your exam B on Wednesday, Wednesday to Thursday, and then that will wrap up the semester in terms of the lectures. And then your final exam is next week in this room. Be there or be square. I'll make up a seating chart so you get an assigned seat. I'll post that in Canvas. Any other questions before we get started today? So the first part of the lecture, what I want you to do is to navigate to the protein data bank on the website that's shown there. So the PDB is maintained by the Research Collaboratory for Structural Bioinformatics. And so it's a repository for protein structures. So protein structure can be determined through a couple different instrumental ways. X-ray is probably the most common, but it can also be done with NMR analysis. So let's go to rcsb.org. And once you get there, you're going to search for this identifier, 1ZT5. If you're doing this on your phone, you might have some trouble navigating things. But flip your phone into landscape mode. That will open things up a little bit. This website's easiest to use on a computer where you can get the full window. So that 1ZT5 is the C-terminal domain of insulin-like growth factor binding protein 1, isolated from human amniotic fluid complex with iron 2. So once you navigate there, we'll walk through how you use this site to answer some structural questions about that protein. So the primary sequence is shown in the middle of the slide here. And again, the sequence is written in the one-letter abbreviations for the amino acids. So you have your C terminus and then your N terminus. Remember the sort of rules for reading that. And then between each letter is a peptide bond, so that's what you're to infer. So we want to use the PDB to learn more about the secondary structure of that primary sequence. Everyone got it? You there? So the 3D structure, when you see it, in your tablet or your phone, you can put your finger on it and move it around. So you can flip it, rotate it. You'll see a couple tabs as well. So within those tabs, you can navigate back and forth, getting different pieces of information. I forget which tab exactly the information on the left here is shown in, but if you click through them, you should get to that. So what you'll find is that the primary sequence is listed sort of at the top. And then everything below is showing you structural features of that visual representation. So again, this green ribbon here what you can see where it's just a line, again, those are amino acids. The whole thing is that sequence of amino acids. But then you get to these arrows, those denote sheets. And then you get to the spiral, that's a helix. So if you don't see one of those major representations, you just see the line itself. There's really no secondary order to it. And the data here in that primary sequence, you can click on those, either the position or the amino acid. And then these different bars and dots and triangles and whatnot, you can click on as well, and it's going to highlight some information for you. Use this screen to find the information. So to answer these questions, I mean, you can probably do it visually and get the right answer most of the time. But really what you want to do is, first question is, how many alpha helices are there? So I mean, this one is, visually you can tell by looking at it, there's only one, but again, if you go to that other screen, do you know how to find that information as well? So it's the pink bar. You can click on it, right? So you click on that pink bar, and then sort of at the top right, it tells you the secondary structure. So again, I would say with your phones, you're probably going to have to do a lot of zooming in and out. So that's why a tablet or a computer is easier to do this. So we're able to click on the pink bar there. And then again, sort of above where the primary sequence is listed, when you click on that, it gives you like a range or something, right? So what are the start and stop amino acids of that helix? So E and S. Does everyone see that? If you don't see it, maybe your neighbor does. They can point you along the right path. How many beta pleated sheets are there? How are those represented? So the yellow box, bar, rectangle, rhombus, trapezoid. So you can click on each one of those, right? And it gives you the little sequence that they're a part of. So can you tell what directions they run? So anti and anti. So that you can look at the structure itself, that green ribbon, and see which way those arrows are pointing. So if they're anti, they're pointing in opposite directions. How many disulfide bridges are there? So there's 3. You see where to find that as well. As you're going down the rows of that, I think it says disulfide bridges. How is that represented?
So like it's brown lines with dots on either end.
So a dot, a line, and then a dot. And then yep.
How could you tell if it was anti-parallel or parallel?
If you go back to the structure and you find where those like fat arrows are, the arrowheads are pointing in opposite directions.
So that means it's
Anti. Between what 2 residues does the first disulfide bridge occur? So A is the correct answer. Are you able to find that information? So find the row with the disulfide bridges. And then as you're reading left to right, you're going to click on the visual representation of that. It's a dot, a line, and then a dot. Everyone good? So where is the first buried residue? What amino acid is it, and what number in the sequence? So position 33, and it's proline. So when you click on that, it should highlight it in the structure, right? Is that correct? So from my recollection of doing this, it's about right there. That seemed familiar. So even though this is a target problem, where is the CHE256 residue that starts the second disulfide bridge?
Position 46.
So if I didn't know where it was and you had to look at the structure, could you point me in a region where I could find it?
So it's right where like the second arrows meet.
So these sheets, so right at the arrowhead here,
Yeah,
if you have it oriented the way I do.
correct.
So the CHE256 is right about here somewhere. And so our last question, what amino acid is in position 47?
Is it?
Glutamic acid.
So GLU, GLU.
How do we get E to the
Do what?
I see an E here. So how do you see the G? G.
Is that, I don't know, is that the one letter abbreviation for it?
Yeah, but E is glutamic acid.
Huh?
E is glutamic acid.
Yeah. So G is glycine, right? So there's some of them I remember, some of them I don't. So, you know, most of them will start with the first letter of the amino acid, but some of them have to be different.
Yeah, G is glycine.
Yeah. Question?
Where do you find that? I can't find it.
You have to look at the row with the primary sequence, and then you can click on those amino acids, and it'll tell you the position. So if you spend some time exploring the PDB, what you'll find is, you know, some of these proteins have multiple entries, and that's based on the experimental condition in which the protein is isolated. If there's any other additives in there, what the instrumental technique was. So for example, in X-ray crystallography, it can be done at different angstroms. And X-ray is really like a picture. So it's a static image. But what it's representing to you is the probability in space of finding those elements. So the smaller the resolution, the better. Like if you have an X-ray structure with a large atomic resolution, it's saying somewhere in this region there's that atom. But as that resolution decreases, the space in which you find that becomes smaller, meaning that the structure is better refined. Additionally, solution state NMR is also used. So when you're in solution, there's obviously more degrees of freedom. And so you can see the nature of the non-covalent interactions, such as hydrogen bonding, a little bit better based on the chemical shifts. So there's things that, you know, X-ray is giving you a more visual picture, whereas NMR, you're looking at an actual spectrum and you have to do a little bit more inferring from. So that's the end of that exercise, introducing you to PDB. but it's a resource that you can use now and in the future, as well as that Baychem website, which is nice for, say, smaller sequences. You know, when you get into large sequences, there's probably specialized software that will help you analyze those things. So what we'll do for the rest of today's lecture and then continue on with Wednesday is start looking at DNA and RNA and what the purpose of those things are in relation to the stuff that we've learned so far. And so I want to preface this just with a little bit of history. So the question is, where do we come from? Why do we exist? Is there anyone out there? So life on Earth is hypothesized to come from this abiogenesis. And so that states that life has arisen from non-living matter, such as organic compounds. So we've seen that amino acids, are the collection of the structural component of a carboxylic acid, an amine, and a side chain. And so carbon, oxygen, nitrogen, sometimes sulfur, those are not living, correct? But when you sequence enough of those amino acids into a long chain, that becomes a peptide or an enzyme, which carries out chemical reactions in the body. So that's what an enzyme does. It's carrying out a specific function. Again, it's still not living, right? So where do peptides come from? They're made from RNA, and RNA is made from DNA. So none of those things are living. Would we all agree on that? So how are we in this room living if we're made of things that are not alive? And so that's what this hypothesis is. So evolution is meant to encompass the environment, so Earth, by which the synthesis of organic molecules, so that's the amino acids, the carbohydrates, molecular self-replication, self-assembly, autocatalysis, and the emergence of cell membranes has allowed for the transition of non-living matter to living entities. So that's the scientific definition of evolution. So again, within those words are organic chemistry, functional groups, and then the self-assembly and replication, that really falls upon molecular recognition. So all of this stuff we learned about functional groups, how these elements are connected, what bonds they use, and the non-covalent interactions really have determined what is happening here. So a nice experiment to help shed light on this was the Miller-Urey experiment. So Urey was the principal investigator, and Miller was a graduate student that worked in his group. So you can read the history of Urey. He was awarded a Nobel Prize for the isolation of deuterium. And so he worked in nuclear physics. And so Miller joined his lab at the University of Chicago and wanted to design an experiment that could test this hypothesis of abiogenesis. So what he did was design an apparatus, which we'll show on the next slide, and pumped in some common feedstock chemicals in an experiment to mimic prebiotic Earth. So this is a schematic of that experiment. And so it's primarily a glass apparatus. And let me point some things out here. So in this lower left flask, there's water. And so that's meant to represent oceans. And so that can be heated. So up into here, and then it gets up into here, which is the atmosphere. So water is our oxygen source, and then in the atmosphere, we have methane, that's the carbon source, ammonia, which is the nitrogen source, and then hydrogen gas is the hydrogen source. And so within this atmosphere are these electrodes that will spark to mimic lightning. And so that's providing some energy. So we're going to do homolytic and heterolytic bond cleavage, form reactive intermediates, and then they'll combine. And so below that is a condenser. So that's going to cool things back down into the aqueous form. They'll pool in here. And then from there, you can start draining out samples at certain time periods. So in essence, it's other than having a glass blower make that piece of apparatus, which is pretty complex, the thought is very simple. I'm going to take these four things, sort of mimic the conditions of prebiotic Earth and see what happens. And so taking an aliquot out of that and then running TLC, so you all have done that. What was found, and so let me explain this TLC because it's maybe a little bit different than what you're used to. So in this direction, the eluent was phenol with ammonia. So that's the solvent that elutes the plate. The plate was taken out, turned on a different axis, and then that direction was eluted with butanol and acetic acid. And so this is a picture from the paper. And what you see are those dark spots. Those are amino acids. And so they were able to identify them relative to standards that were run under the same TLC conditions. So what you see is, aspartic acid, glycine, alanine, beta-alanine, and alpha amino metabutyric acid. So notice that none of those things went into the flask, right? It was hydrogen, ammonia, water, and methane. So from those four basic feedstocks, these amino acids were produced. And people have subsequently repeated these over the decades under fancier conditions, and they will produce all 20 proteinogenic amino acids. And so I would assume if you let that cook long enough, those amino acids are going to start to couple into dipeptides, tripeptides, and eventually it would form an enzyme, right? And so it's interesting to think about, your own physical and chemical composition in terms of this experiment. So there was a New York Times article that kind of recalled this experiment. And those are the actual vials of the materials isolated. And as instrumentation has evolved over the decades, people went back and isolated those samples and ran them on fancier instruments and found that there were actually more amino acids than they were originally able to identify. So the statement here is you're almost done. We have 45 minutes today, 75 minutes Wednesday, and you're almost done with organic chemistry. But really that sets the stage for moving on and understanding how these large biomolecules function in life. So understanding the key structural components will help you better understand what they do and why they do it. So after that experiment was published, Feynman wrote a poem. So Feynman was a Nobel laureate in physics, and he was one of these people that was interested in a lot of things. He played the drums, he was an artist, that sort of thing. So he wrote this poem, and what I want to highlight is the last sort of two sentences here. So I, a universe of atoms, an atom in the universe. And so at this point in human history, based on quantum physics, humans started to understand the nature of the atom and ultimately into living organisms as well as like the universe itself. And so all of it really gets back to how elements in the periodic table bond to each other and how from those simple rules we can make complex things that ultimately become us. So again, to put it into perspective, so for you to exist, each generation, assuming a 25-year reproductive cycle, requires 2 to the n ancestors. So you would be sort of line 0 here. Then you have parents. So that's 2 generational ancestors, so 2 total. And then from there you have 4 grandparents, so that gets you up to 6 generational ancestors. So you can go back to the 10th generation there, and it requires 2,046 human beings have existed to make you. And what's interesting to think about, look at the span of those those dates of birth. So your 8th great grandparent was probably walking the earth around 1750, and they did not have iPhones. They did not have cars. So each generation there's been, these advances that get us to where we're at. And if you track over the years, the lifespan increases, right? So, back then people lived to 18 or something. I mean, you had your kids worked in the field or whatever, and then you died. I mean, because you didn't get enough food, you got sepsis, and then, you got some disease that couldn't be cured. And so as, science and medicine has evolved, it's allowed us to live healthier, more productive lives, but also to pursue why things happen, right? So I relate this to my kids. This is my favorite Dr. Seuss book to read to my kids. You have brains in your head, you have feet in your shoes. You can steer yourself in any direction you choose. You're on your own and you know what to do, and you are the one who will decide where to go. So college is tough, but again, you really have to find out who you are and make those prior 8 generations, 10 generations proud of where you've become. So with that, I'm going to switch over and we'll watch a little movie on DNA, and that'll set the stage for understanding DNA, how it makes RNA, how it makes peptides, proteins. So we're going to just walk through that again. I mean, it pretty much laid out all the information that I'm going to tell you now, but we'll go back through the different structural features. And ultimately, what we want to understand is how DNA is used as a language to make RNA, which makes proteins. So DNA is a biomolecule responsible for carrying genetic information, and it's stored in every cell. So here are some facts. So for a human child, female and male, at those sort of average weights, there's the average number of cells. So we're into what millions, billions, trillions. Looks like about the billions, right? So how does DNA get stored in a cell if we have so many cells? So the average DNA strand is 2 meters long. So it's like 6 feet or something, right? So we're considering DNA now as a molecule. So like from one end of the strand to the other is 6 feet long. And so, what's your height, multiplied by the number of cells by 6 feet. I mean, all of that stuff is contained within your body. So what actually happens? So the helical interaction, you can take something long, and if you start coiling it, that makes it shorter, right? So probably none of you will say yes to this question, but they're like, when I was growing up, every house had a telephone on the wall, and that telephone had a wire. I mean, like, you don't have that now, right? So the wire was usually coiled. And the reason for that is because then you could walk into the other room and it would be longer. So that's an example of, taking a length that's long, you coiling it, it makes it shorter, but you can still get that ultimate length. So that helps conserve on the space a little bit to an extent. But another thing happens, those coils actually start to then coil. So there's a protein called a histone that will interact with DNA and coil it even further. So as you saw in the video, that orange blob sort of coming up to the DNA, that would be like a protein, or if you consider it a histone, it's going to interact with it a certain way, and that's going to help that coil. And so these strands start to occupy less distance and volume, and eventually the DNA superstructure becomes visible under a microscope, and we call those chromosomes. So, the chromosomes kind of look like those, I don't know, like little men or something, like kind of like that or whatever. So that's actually a DNA molecule that's super coiled. And so on Wednesday, we'll watch some more videos where they sort of show that process. So chromosomes are supercoiled DNA that encode genetic information. So now keep in mind that the DNA is made of 3 structural components: a sugar, a base, and a phosphate. Those are not living, right? The sugar is the same, the phosphate's the same, but there's 4 bases, so A, T, and C, and G. Within that is the genetic information that makes you a living person. And so, if that strand is 6 feet long and you have 4 different bases, you get all of these combinations that make your eyes blue, your hair brown or blonde or whatever, your height, your weight, all of that stuff. So portions of DNA are called genes that code for the formation of proteins. So essentially what happens is a protein comes along, it reads it then makes RNA. The RNA then gets turned into peptides. So let's focus on RNA first. So RNA stands for ribonucleic acid, and the ribo comes from ribose. So we saw that in the carbohydrate section of the lectures here. And so ribose is an aldopentose, and so we're using the natural sugar, so it's the D configuration. And what you see here are the 2 anomers. So that's the anomeric center. And the number 2 is telling you the oxidation state, where that carbon is equivalent to an aldehyde. So it's basically a secondary alcohol is trapped the aldehyde to make the ring. The anomers tell you the up or down position of the alcohol at that position. So alpha was down, and then beta was up. So the other thing I want to point out, again, it's a 5-membered ring, and those are called furanoses. Your typical 6-membered ring like glucose, those are called pyranoses. So the reason I want to start with RNA first is that it's the fully intact ribose molecule. So when we get to DNA, I'll re-explain what that D means. So the 3 structural components. So you have your heterocyclic base. Again, that can vary. It can be 4 different things. That base is bonded at the anomeric position. So notice the heteroatoms bonded to it now are O and N. For organic chemistry, we don't call that an acetal, it's called an aminal. But it's at oxidation state 2 with an sp3 hybridized carbon. Remember that acetals or aminals were acid labile. So with treatment of acid, it will go back to the sp2 hybridized carbon. The sugar is ribose, so that's the same no matter what. Remember, it's the base that changes. So the 3-position alcohol gets bonded to an oxygen from the 5-position phosphate. So what you're looking at is a monomer of that biopolymer. So your beta D-ribose, and that's linked by phosphate. So as I said, we'll go through these structural features, and one of the things we'll eventually look at here is why phosphorus there. Why doesn't it just link through an alcohol or some other functional group? So we'll look a little bit into the location of phosphorus on the periodic table, what's special about it, why nature has chosen it. So the base, so it's a nitrogen base. Again, it's bonded to the anomeric center. So right now, you'll see the difference between RNA and DNA is a modification of one of these bases. But essentially, they're the same 4 things. So we have cytosine, uracil, adenine, and guanine. And so the 2 classes, I'll draw a line here. To the left, those are called pyrimidines. And to the right, these are called purines. So those are the molecules themselves. So cytosine, when it gets incorporated into the sugar, the name changes a little bit. It's called cytidine. And so you see that modification for each. That's telling you kind of what structure it's in at that point. And as the video said, when you look at them, the molecules are essentially flat due to the hybridization of the atoms involved. And that flatness allows for these hydrogen bonds to form. And then additionally, as we'll see with DNA, because they're aromatic, you can get this non-covalent interaction called pi-pi stacking. So now these are the nucleosides. So again, pay attention to the nomenclature. The nucleosides are the molecules shown. When they get incorporated into a chain, then they become nucleotides. So the numbers in red, again, are the oxidation states according to, counting the carbon bonds to the heteroatoms. And then you identify each of those individual functional groups, and essentially what they are hydrogen bond donors or acceptors. So I think let me ask you, from RNA to DNA, which one of these changes? What? The uridine. So the uridine changes a little bit. It gets a methyl group on the heterocyclic base there. So to understand why these things hydrogen bond, which nitrogen atom is the most basic? Which one? So A, B, C, or D? So 3 of them are kind of the same, one is very different. So B. So when we look at A, C, and D, all of the nitrogen atoms are sp2 hybridized. For A, C, and D, the lone pair resides in a p orbital. And that lone pair is then delocalized into the carbonyl through resonance. But for B, notice that it already has a double bond. So its p orbital is already tied up. So the lone pair is in an sp2 hybrid orbital. So that's the nitrogen atom that's going to hydrogen bond. So that'll be the acceptor. So in uridine, which hydrogen atom is the most acidic? Yeah, D. So how do we know that? B and C are both alcohols. They have a pK of around 16. A is bonded to an sp2 hybridized carbon, so it's like an alkene. Its pK is around 40, 45. D is bonded to this nitrogen. And so if you deprotonated that, the negative charge could be delocalized into that carbonyl or that carbonyl. So that stabilizes the resultant base, therefore making that the most acidic proton. Meaning if it's the most acidic, it's more likely to participate in hydrogen bonding. And so that analysis will help you understand how these things come together in those ratios they do. So let's talk a little bit now about phosphorus. And so why is phosphorus used at that 5' position? In the periodic table, phosphorus is directly under nitrogen. So at that point, it's in row 3. And because of that, it has access to d orbitals. The d orbitals allow it to attain geometries that you can't normally, like with period 2. So if you remember from Gen Chem, those d orbitals are listed here. And then you can look at phosphoric acid. So if we want to generalize that phosphate group, we can generalize it to phosphoric acid. You can see the oxidation state is 5. So normally, that's not attainable with carbon, right? So by moving from nitrogen to phosphorus, we get this geometries that you can't obtain due to the d orbitals, and then a higher oxidation state. So when you look at that, it has 3 sigma bonds to oxygen and then 1 pi bond to an oxygen. And so phosphoric acid itself is pretty interesting because it can exist in a lot of different forms. The first thing we'll talk about is the pKa's, because it's triacidic, right? So H3PO4. And as we've seen with the amino acid analysis, when we're doing those titrations, starting, say, from 0 going up to 14, when you have multiply acidic functional groups, you can get different pKa values. So for phosphoric acid, we have 2.2, 7.2. So that's right about where we need to be under physiological conditions, and then 12.7. So if we analyze this from left to right, so there's phosphoric acid. We're under acidic conditions. If we're titrating in base, after we hit 2.2 and then we're increasing the pH, we're going to do a monodeprotonation. So we end up with a single negative charge there. Now look at the next span. So from 0 to 2.2, that's 2 pH units, 2.2 pH units. Now the next span is 5 pH units. So it'll exist in this negative form until it hits 7.2, and then once you surpass that, you do the second deprotonation. Then it's a dianion. And then you're going to hit 12.7, you do the third, and that's when you get the phosphate. So now it's the trianion. So these things are, if you're trying to buy them, they're referred to different names, like hydrogen phosphate, biphosphate, triphosphate, that sort of thing. And then you can stick any sort of metal that you want around there to balance the charge. Has anyone ever worked with phosphoric acid in lab? So the pain about it is it starts to do all these other things like dimerize, trimerize, and whatnot. So phosphoric acid itself is also referred to as orthophosphoric acid. But just sitting around, it can form an anhydride by losing water. And so this is called pyrophosphoric acid. Now, if you lose another molecule of water and add a third equivalent, now you have tripolyphosphoric acid. So does that look kind of familiar to anyone? From your biology courses? So ATP, what's that stand for? Adenosine triphosphate. So what happens is within that part, it's kind of your energy source and reserve. So you can start adding these things, clipping them off, and that supplies and takes in energy to get through metabolic cycles. So the tripolyphosphoric acid is also primed, decyclized by losing H2O, and this is called trimetaphosphoric acid. So phosphorus is pretty versatile like that chemically, and that's been exploited through evolutionary processes. So one interesting kind of side note to RNA is that the 2023 Nobel Prize in Medicine was awarded for mRNA vaccines. So I'm going to raise my hand, but no one in here, don't raise your hand. So I got the vaccines, I got boosters. Up until a couple years ago, I just got lazy and didn't do it anymore. But so the COVID vaccine is based on these mRNA science. And so Carrico and Weissman were awarded that for their discoveries concerning nucleoside base modifications that enabled the development of effective mRNA vaccines against COVID-19. And if you read the story, it's pretty interesting, especially with Katlin Carrico as a female scientist in STEM, and we'll see a little bit more about this on Wednesday about the biases that exist in scientific research. She had been working on this for decades. This was not something that, hey, COVID happened, let's go in the lab and make a vaccine. So the thought process of doing these modifications has existed for a while, and then this pandemic happened, and then the use for it was found. So what did they do? So look at the structure on the left, and then look at the structure on the right before I start pointing anything out, just verbally. So focus your eyeball on the anomeric carbon and see if you can understand the modification that they made. So my hope would be that through my teaching, you'll understand just in a way, a simple modification chemically allowed them to produce these vaccines on a large scale. So as I said, the anomeric carbon so that's acid labile because the oxygen atom can push that group out. And so in general, that makes strands of mRNA unstable. So essentially what they did, and I'll highlight it in, or draw it in green here. So they got rid of that instability from the anomeric center. So essentially, they took that position, moved it up there, took that carbon and moved it down. And so now there, it's a carbon. So it's no longer at an oxidation state of 2, it's at 1 and it's more stable. Another way you can think about it is they just took this off, flipped the molecule around and bonded the carbon atom there instead of that nitrogen. And so if you think about just the functional groups, the reactivity, it's a pretty nice application of that. And then, I do challenge you to think when you're in lab and you're running a reaction on 50 milligrams or whatever it is, tiny scale, what if you had to make a shot to give to like 4 billion people? How are you going to produce the amount of material that's needed? And so these kind of structural features, analysis and design help meet that need. And I would be pretty confident that, even though they're biochemists, virologists, immunologists, they were thinking back to organic chemistry as the rationale for doing that modification. So I think what we'll do is we'll stop here at DNA, and then on Wednesday we'll continue on with DNA, the differences, the two position. We'll watch a couple videos, and then we'll see ultimately how DNA makes RNA makes proteins.